This is Mark Strassman, reporter with Etopia Media Medical News Network. We're about to speak to Robin Williams, who's in the biology department at University College London. I'm in Los Angeles. Welcome to Etopia Media Medical News Network.
Thank you, Mark.
Tell us just first where you do your research and who funds it.
Okay, so I'm based in the biology department at University College. I'm funded by the Wellcome Trust, who's a basic and clinical biological research funding charity. So they fund research to basically help understand disease, I suppose.
Okay. We could do this in one of two ways. Maybe we'll do it in both. Tell us when manic depression became bipolar disorder, and whether that marked any significant step in the evolution of the treatment or understanding of the disease, or whether it was mainly a semantic transformation.
It was mainly semantic. People out there in society think that manic depression and bipolar disorder are different things. They are basically the same disorder. What confuses people is manic depression. People associate that with depression. Now, that's not the case. Bipolar disorder more accurately reflects the actual condition. So people go through stages where they are in a manic phase, and then they go through a depressed phase. Now, clinically, it's very different to depression.
The biochemical state of people's minds when they're clinically depressed versus when they're in a depressed state of bipolar disorder are different biochemical status?
Yes. So the drugs used to treat depression are very different to the drugs used to treat bipolar disorder.
Okay.
Although many patients are often put on a combination of them, the most common bipolar disorder treatments, lithium or valproic acid, are specifically for bipolar depressed patients or manic depressed patients.
Okay, we're going to talk about that more. Tell us how and why you got interested in studying bipolar disorder.
I did my second postdoc about 8 years ago, and I was looking at lithium. But we started off looking at lithium to look at targets in the cell, the proteins that are affected by lithium. Some of our research then identified an enzyme which was variant in bipolar disorder people. So this led to a greater understanding, basically, of how bipolar disorder drugs work.
Okay, before we talk about how the drugs work, let's talk about the disease itself. What are the symptoms and the general form that the bipolar disorder takes, and how prevalent is it, and how debilitating is it?
Well, I'm not a clinician, so I don't work directly with bipolar patients.
Right.
What I do is basically look at the drugs, bipolar disorder drugs, and I put it on cells, and I work out the biochemistry of how these drugs function. So in that way, we can extrapolate that to look at people.
Okay. Was this kind of a breakthrough? Was lithium used for a long time, more or less serendipitously, and people didn't really know what its mechanism of action was? Yes,
that's an interesting story. Lithium was initially found in the late 1800s as a treatment for melancholia. Now, they originally started using this because they thought that gout, which is a buildup of uric acid in your extremities, could be cured by lithium treatment. It turns out that the salt for uric acid is very soluble if it's the lithium salt. So people were given lithium to try and dissolve the uric acid in their extremities to get rid of gout.
Did it work?
Now, they found out accidentally that it was also functional in treating melancholia.
Did the lithium work as a treatment for gout?
No, not that I'm aware of.
But it did have an effect on a bipolar patients.
Yes.
And
so at the time, I think they gave a dose of something like 5 crystals of lithium per day. So we've developed a long way from there, obviously. But the history of lithium is it was used initially around the turn of the century and quite successfully, but then people began using it more and more. Now, lithium is quite a toxic substance, so if you're given too much lithium, you might die eventually. One of the most famous uses of lithium was in a drink called 7-Up, a lemonade soft drink, and lithium was initially put in 7-Up. I think then in the 1930s
in the same way, I suppose
taken out.
Cocaine was put into Coca-Cola.
Yes, exactly. Yeah, I think opposite effects somehow.
Right. People drank
so
7-Up. Yes.
Lithium went through a bad patch because it killed a few people,
right.
And now people are very careful on the dosing they give for lithium treatment.
Okay, talk about valproic acid. What is it, and how did it come to be used as a treatment for bipolar disorder?
Okay, so valproate is 2-propyl pentanoic acid.
Right.
It's a short-chain fatty acid. Now, again, this was found accidentally. The history of that is people were designing new treatments for epilepsy. Now, they found that it was very difficult to dissolve the anti-epileptics. So one scientist had 10 different derivatives of valproic, had 10 different compounds that he wanted to test, and he couldn't dissolve them. So he found valproic acid worked very well to dissolve all of these 10. So he injected mice, epileptic mice, with the 10 compounds dissolved in valproic acid, as well as valproic acid as a negative control. And from these experiments, he found that all 10 drugs and the vector control worked well to stop epilepsy. So that was a great breakthrough, but it was, again, serendipitous.
And is valproic acid still used in the treatment of epilepsy?
Yes, very widely. It's, I think, the second or third most commonly used treatment.
And how was it found then to be effective in bipolar disorder?
Like all the current treatments for bipolar disorder, it was just given to a population, some of which had manic depression, and they showed marked improvement. Okay,
so, but when you came along, there was really no clear elucidation of the mechanism by which lithium or valproic acid worked in bipolar disorder, was there?
Well, there was a theory which was called the inositol depletion theory, which was based on the function of lithium in the cell, where people found that lithium stopped this signaling cycle, chemicals in the cell which would be recycled, and lithium would slow this recycling. So this signaling pathway, it was thought, would be overactive, and you give a patient lithium and you slow that recycling, so you slow the overactive signaling mechanism.
So it would block the reuptake of the inositol?
More the recycling of inositol within the cell.
All right. And what's the status of that theory now?
Our data supports that very well. We find that lithium, valproate, and carbamazepine, which are the 3 most commonly used anti-manic drugs, all function to modulate inositol signaling.
All right. Now, do these 3 drugs provide a sufficient selection of agents that work effectively on bipolar, or do they have serious deleterious side effects?
They all have different side effects, yes. My work is mainly on valproic acid, and that has quite considerable side effects.
What are they? Yes.
The most important really is teratogenicity, which is if a woman who's pregnant is taking valproic acid during her first trimester of pregnancy, she has a small increased chance of getting birth defects. So pregnant women taking this have to be very careful when taking valproic acid.
Okay. So what is it that you set out to investigate then in your study of valproic acid, and where does the slime mold come in?
Okay, so we were trying to find out the signaling pathways and the genes and the proteins involved in how valproic acid work. So we looked at various derivatives of valproic acid, thinking that it was inositol depletion, and we were able to find certain compounds which would act well to deplete inositol, but didn't have the teratogenic side effects.
Right. And what have you found?
Well, we found two compounds which we think are very good in inositol depleting, but don't have the teratogenic side effects.
Do they have?
So this would mean that pregnant women taking an anti-manic treatment would be able to take this safely. If the drugs go through clinical trials, then they would be able to have this, and they wouldn't have to worry about pregnancy.
Tell us about the slime mold and how that fits into the research.
Well, the slime mold is a very good model system. The National Institute of Health has labeled dictyostelium discoideum, which is a cellular slime mold, as a model system for biomedical research. So they realize that we can look at this slime mold and try and find out more about medical problems, really. So we employ dictyostelium as a good model system to try and work out what happens with drugs and with proteins in the cell and how they're related to medical problems.
Okay. Talk a little bit about the genetics and the proteins involved in bipolar disorder and in valproic acid's depletion of inositol.
Okay, we know how lithium works. We know two proteins in the cell which are involved in inositol cycling. So we have the exact target of lithium in this cell for inositol depletion. Valproic acid, we don't know how it works. There was a recent paper a year ago that found that valproic acid slows down the biosynthesis of VPA within the cell. Sorry, slows down the biosynthesis of inositol in the cell. But exactly how it does that, we don't know.
So you've got it down to that molecular level, but you don't know the mechanism yet by which the valproic acid does it. Do you have any hypotheses about how it interferes with the pathways to make that happen?
That's still being researched. We're still looking at that as quickly as we can. We have more papers and data being prepared currently, which I think is going to shed more light on it. But until those papers go through the publishing process, we're not really sure.
All right. What benefits would accrue by understanding the molecular basis of the effect of valproic acid?
Well, anti-manic treatments aren't always 100% functional. There are a number of people out there who have manic depression who aren't completely cured by the treatments. Those that do benefit, I suppose, by the treatment might still have bad side effects. So if we can understand how the drugs work or understand the proteins that are targeted by them, then we can get better treatments and safer treatments for the people in society that have bipolar disorder.
Are there genetic markers or known genetic factors involved in bipolar disorder?
That's another area that's being hotly pursued. There are a number of loci, a number of genes which are common to bipolar disorder families, and there's a big area of science pursuing that, trying to find the genes that are passed from parents to offspring which will cause an increase in likelihood of getting bipolar disorder. That research is still continuing, but we haven't got any firm genes which are responsible. But what we do know is there are many genes.
All right. Now, what's the relationship between the search for the genetic factors in bipolar disorder with the search for the mechanisms by which the proteins operate to cause it or to have it stopped with the treatments?
They're complementary approaches, really. What we need to do is find how the drugs work in the cell and then look at people to see if that's how they function therapeutically. But we also need to find the genes in people being passed from parent to child to be able to identify whether a person has bipolar disorder, what type of bipolar disorder, and how we can best help them not experience the ups and downs that bipolar disorder causes.
So when these two streams of research come together, the idea would be to understand what genes code for what proteins in what sequence and proportion that generate the symptoms of bipolar disorder, right?
Yes, perfect.
Okay.
Yeah.
All right. Could you set a timetable when you think that's going to happen?
My father, who's Australian, and I speak to my parents every fortnight. My father sweetly asks every now and again, "So have you cured manic depression yet, Robin?" Okay. I have to laugh and say, hopefully by the time I die, we will have a better understanding and we might be able to stop bipolar disorder completely.
Would that
it's
involve...
a long...
Yeah. I'm sorry.
Sorry?
Would that involve germline manipulation?
That would be an ethical decision that society will have to make when the technology is available.
But that's essentially what is being made possible by this research, isn't it?
Given that would be a possibility, yes.
Okay. And do you think I understand that you work more on the biochemical than the clinical level, but do you think that would be a good thing to correct this biochemical imbalance and eliminate it in individuals, or generally, is there any evolutionary purpose that you see that's been served by the existence of bipolar genes?
Again, that's quite a hot issue. There are a number of people in society that have bipolar disorder, in the past at least, who have been fantastic, renowned artists and creative thinkers, possibly due to bipolar disorder.
This is what Dr. Redford wrote a book called Touched by Fire, which deals with this.
So we have to, as a society, think, well, do we want to eliminate this disease and possibly not have 1 in 10 million geniuses? Right.
Is there a relationship between genius and bipolar disorder?
Only that a number of people in the past that have been called a genius also had bipolar disorder.
All right. But there's obviously, or is there any genetic? You can have one without the other. Obviously, you can be bipolar without being a genius, and a genius without having bipolar. Is there a higher percentage confluence of these two conditions?
Oh, I'm not sure that's been done, really. Right. But what I would say is the majority of bipolar disorder patients will go through their manic period and they would feel intelligent, creative, and they could save the world. And they can do great stuff.
Well, do they feel that? Do they feel that way only, or are they capable of actually making contributions that would save the world, or parts of it?
Well, those geniuses in the past that have done this obviously have added benefit to our world. So. All
right. So it's a tricky question, then, you say.
Yeah.
Okay. Why don't we wrap up by focusing on the more down-to-earth timetable for your research and what kind of benefits you expect to be derived on what kind of timetable, and when people with bipolar disorder can expect, especially as you've mentioned, I guess pregnant women can look forward to some specific benefits being available.
I'm unfortunately a research scientist in a university, so I can come up with a model and design new treatments and go, look, here is a new drug and I think this would be beneficial. It would be up to major pharmaceuticals to go, okay, this looks like an option, we need to do further toxicity tests and then pilot studies and try it on people. that will take years, many years.
Are any of those trials underway yet? And you said your research is funded by the Wellcome Trust. They're a pharmaceutical company. Have they shown any interest in commercializing your research?
No. The problem I find is to get a patent, which would be the first step in this, we really need to define the whole family of drugs which will do this. If I apply for a patent, someone may go and make a small variation on the compounds I'm applying for,
right,
and overcome that patent. So I'm pursuing this work with various international colleagues, and we will hopefully, in a couple of years' time, come up with a family of compounds which we can say these compounds will possibly function in manic depression treatment, and from that we can then go find ones which are non-toxic and then go to clinical trials.
So you're saying you need to be able to define the entire range of agents that can do this, in part so you can get the patent or patent to cover them, and then start selecting ones within that group for further tests.
Yes, that's right.
Okay, good. All right. Would you say finally that the enjoyment you take in this work is more on the understanding of the biochemical mechanisms or the possible good that's being done, or is there from them, or is there a balance there for you? Where do you get your greatest satisfaction as a researcher?
I feel delighted that I can go out and speak to members of the community and go, my work will benefit the community. I think that's a very good thing, and I'm delighted I can do that. I love science. I think I'm a scientist in my heart, and I am just very lucky to be able to pursue an area of research that I think can directly benefit society.
All right, good. I want to thank you for doing the work and for explaining it to our listeners and viewers today.
It's been a pleasure.
Thank you.
Thank you.